My name is Avantika. I am a fourth-year graduate student. We're here in the McCune lab at San Francisco General Hospital, and I'm part of the Biomedical Sciences graduate program at UCSF. Specifically, I'm actually working on intestinal epithelial cells, which are the cells that line your intestine essentially and keep it basically form a barrier that will prevent anything that shouldn't be inside your body from actually being inside your body. And I'm working specifically in the context of HIV infection and progression to AIDS disease. And the whole idea is that intestinal epithelial cells are really important in that scenario because the gut and the immune system that's in the gut is actually really important for HIV and is one of the most affected areas for HIV. And the intestinal epithelial cell barrier that I'm working on is really important in making sure that there's not damage in the intestine. And a lot of damage happens just kind of by a factor of HIV and the virus itself. But it's really important to keep that barrier so that you prevent any excessive damage and you can try to get repair and also promote kind of healing from the disease and prevent any kind of excessive side effects that would happen. So I actually grew up here in the Bay Area. I grew up in the South Bay in a city called Los Gatos. My mom actually started as a pediatrician and then kind of early in my life decided that she was going to transition to research and actually doing plant-based medicine. My dad, on the other hand, is kind of an entrepreneur and has started a number of small different types of companies with different focuses and currently is focusing on solar. I had a wonderful teacher in high school who really taught me that it wasn't just about science and learning about science, but it was about the responsibility that you have if you go into science, and that really drove me into the kind of research that I do now, which is translational based or medicine based research, therapeutic based research rather, and trying to understand human disease as specifically as we can, and then trying to find ways to prevent human disease from happening or from resolving it if it actually occurs. The blood separates out into the different layers, and then your cells are kind of at this interface right here. So I actually chose this particular lab based on its focus on community health and disease and its emphasis on translational medicine. And I didn't come into UCSF knowing that I would do HIV research, but when I got here I realized that it was something that I was very passionate about. And the community feeling here at San Francisco General really is amazing because it was one of the centers of the AIDS epidemic in the 80s and has been built up to be this tremendous cohort of patients and clinics that we're able to work with and doctors and collaborators. So it's a really rich environment, and it sort of just fit for me that this was the right place for me to be to do the type of research that I wanted to do. So a lot of what I do is actually based on clinical specimens, and we receive clinical specimens from the San Francisco General Hospital here. I'll isolate cells out of that and run a number of assays on that. And we get clinical specimens from really uninfected patients and infected patients as well, and trying to make those comparisons and see if we can determine relationships between the epithelial compartment and the immune compartment and how that relationship might change based on your disease status. A lot of the other stuff I do a lot of tissue staining here, which is fun. You get to play around with fluorescent colors and images, and that can be really beautiful. It's a really good kind of visual interpretation of your work. Whereas I think a lot of science can seem like it's a point on a plot, but that's a really fun thing for me to do, and that was actually the first thing I learned to do in this lab, and I didn't know it before I got here, but now everyone calls me the IHC expert, so that's fun. I tend to do a lot of flow cytometry here, so I like to delineate a certain type of cell that I'm looking for or delineate their function, such as cytokine production. So right now I'm basically measuring plasma that we've taken from the blood. And I'm measuring it for certain proteins that are present in there. Now I know the system very well. I understand its intricacies. Obviously there's so much more for me to understand and so much more for me to learn, but it's a system that I understand, and I love how complex it is and how many different components there are to it, and trying to understand all of the interactions, and that's precisely what I'm trying to do, is understand how the epithelial cells are interacting with the immune cells underneath it, both kind of in a normal scenario and how that changes with HIV, and if we can kind of alter any negative interactions that might happen there. So I've always been asked why I didn't go into medical school, and my number one answer for that has been that I think that medicine is really important, and obviously it's a core part of our community, but I think that science informs medicine, and that's, I think, what I mean by responsibility, in the sense that what we come up with as scientists, what we do here on the bench in the lab, will really inform the therapies of the future, and that's really that responsibility that we have, to try to be creative, to come up with solutions that may not be out there yet, or improvements on solutions that are already out there, to try and you know, further improve community health and patient health. It's a social experience, it's fun, it's entertaining, it's creative, it's a game in a way, trying to come up with your project and figure out how you're going to achieve it. I would love to say that my research project informed a therapy that had an effect on a patient community, whether that's now in regards to HIV disease and the complications associated with HIV, or whether that's later, you know, as concerned with my future work. My ultimate goal is to say that I did that work, and that work changed that particular therapy and improved that patient's life. That is 100% my goal. That has been my goal since that anatomy class that I talked about in high school, where I learned that it's not just health, it's health in the community and health in the patient. And I would love to say that I had that impact on someone's life, absolutely.